Hi, John. Thank you for coming and join us today.
Very happy to be here. Thank you.
Yeah. So Delicia is our director at Research Center of Applied Zoology, John.
Nice to meet you.
Yeah.
Nice to meet you. So, can we start right now?
Baik, kita mulai, Bapak Ibu. Ya, selamat siang teman-teman semuanya yang sudah join di kegiatan kali ini. Seperti biasa, kita akan mengadakan sesi regular sharing dengan nama Summer School, di mana reguler ini merupakan kita lakukan tiap 2 minggu satu kali. Kita optimalkan untuk membagikan dan sharing ilmu ke teman-teman, tidak hanya internal di BRIN, tapi juga semua institusi yang memang keterkaitan dengan riset yang sedang dibicarakan, baik itu akademisi, profesional, dan mungkin bagian-bagian lain yang berkenan untuk join. Pada hari ini kita akan kedatangan narasumber. Ada 2 ya, dan yang satu nanti menangkap moderator. Untuk narasumber yang pertama ada Dr. John Bastiansen dari Animal Breeding and Genomics, Wageningen University and Research. Beliau akan membawakan presentasi dengan judul Strategies for Developing Base Population and Enhancing Genetic Diversity in Fish Domestication. Dan yang kedua, narasumber kedua adalah Dr. M.H. Fariduddin Attar. Beliau dari Pusat Riset Zoologi Terapan BRIN dan akan membicarakan mengenai breeding for resilient growth in tilapia. Baik, sebelum kita mulai, seperti biasa, saya akan memberikan kesempatan kepada Ibu Kepala Pusat Riset Zoologi Terapan, Ibu Dr. Delicia Yunita Rahman, MSI. Waktu dan tempat kami persilahkan. Bu Kapus.
Terima kasih, Mas Bram. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Welcome to the Research Center for Applied Zoology Summer School Series 9. It is a real pleasure to see many enthusiastic faces joining us today, colleagues, students, researchers, and friends who share a passion for aquatic animal breeding and sustainable aquaculture. Events like this are precisely why our research community thrives. They bring together diverse perspective, practical experience, and fresh idea that push our field forward. We are especially honored to host 2 outstanding speakers. Thank you to John Bastiansen from Wageningen University and Research for taking the time to give this talk. Although we have like 6-hour differences between Jakarta and the Netherlands. Talking about the Netherlands, I'm familiar with it as I spent almost 4.5 years there completing my PhD in Groningen back in 2018. Given the main task of the Research Center for Applied Zoology Group, especially within the focus group on aquatic fauna and related to the research on domestication of fish, this talk is important to provide guidance and a solid basis for initiating domestication effort. We currently have ongoing research, including the characterization of potential fish population for domestication, studies on fish reproduction, and also population management using genetic approaches, including the application of the LUP. Later, Farid will also give a more complete update about our ongoing research within the aquatic fauna focus group. As a follow-up step, if later that John have any find common research interest, we can also initiate collaboration by joining existing project or by developing proposal together to secure funding for joint research. And for the other participant, I encourage everyone to listen actively, take notes, and engage with our speakers. Do not hesitate to approach the presenter afterwards if you like to continue the conversation one-on-one. Without further ado, let us get started. Enjoy the session and good afternoon. Thank you.
Baik, terima kasih Bu Kapus atas opening remarks-nya. Tanpa berlama-lama, saya berikan kesempatan kepada moderator hari ini yang juga menjadi narasumber, Dr. Farid Attar. Waktu dan tempat kami persilakan. Bisa.
Thank you, Bramantyo. So I am Farid, researcher at the Research Center for Applied Zoology. Today, I will have a multi-role as moderator for this session and also to give us a short talk about the update of my study. So first, I would like to start this first session. Talk from John Bastiansen from Animal Breeding and Genomics, Wageningen University and Research. I will introduce you a bit, John, about you to the participants. So John is please correct me if I'm wrong. So John is a research associate at Animal Breeding and Genomics, Wageningen University and Research. Within Animal Breeding and Genomics, we have John is joined with the focus group of aqua breeding, so people that work with the fish breeding. John's research focus is on aquaculture breeding program, genomic selection, and also quantitative genetics. Most recent work in 2025 centers on integrated deep learning derived image phenotyping and GBI analysis to advance aquaculture genetics, and especially in aquaculture species like trout, sea bream, and kingfish. And also his projects also span economic modeling and the development of resilience and tolerance traits and in tilapia and several aquaculture species. Without ado, John, I would like to give the floor to you. You can start to share your screen and start the talk. Please, John.
Okay. Thank you. You mentioned a lot of things. Some things are more recent than others, so not all of that is happening at the same time. First of all, I'd like to thank you for the invitation. I always enjoy presenting about our work. We have a connection. Let me share the slides. We have a connection from our group to several places in Indonesia where we have done collaborative research and also ongoing research on bioeconomic modeling of polyculture, tilapia shrimp, which is currently ongoing with our PhD student, Melissa. Share this one. Do you see the slides?
Yes,
Okay, good. I kept on this slide a few nice pictures that we took during my most recent trip to Indonesia. This is taken in the Brebes area on the north coast of Java, where we 2 years ago were running an experiment on polyculture, keeping tilapia, red tilapia, as you see here, together with seaweed. I'm not going to talk about that today. I'm happy to talk about that on another occasion, or we could invite one of our colleagues that is an expert on seaweed, not fauna, so maybe for a different group. Go here. Okay, so I'm part of the animal breeding and genomics group in Wageningen. Some of you may be familiar, but I'll explain that. So Wageningen is a combination, and our group is also a combination of around 100 people, equally divided between the university, which has the duties of teaching and research, and the other half of our group is employed by another entity, Wageningen Livestock Research, which is a research organization in some ways similar to BRIN, So it has duties, government duties, research duties that are being performed by our group, and we also do consulting with or R&D with companies or with other institutes. So it's contract research and government tasks. You see that we're a big group. There are some familiar faces if you zoom in, probably here in the middle of the group. The organization of the group is like this. There's something in the middle of my screen. Hide video panel. No, that's not what I want. Sorry. Okay, the meeting controls were in the middle of my screen, so which was distracting me. This is the structure that Farid already mentioned. Our group is divided over focus groups. At the top you see aqua breeding, which is the group that I'm part of. In my group we work on a lot of aspects of breeding, and other focus groups, they are more technology focused. So you see there's a group that is focused on genetic diversity, which we collaborate closely with, but also on laboratory techniques, DNA analysis, genomic prediction is a big research topic also in our group. So as an aqua breeding group, we are able to collaborate directly with colleagues that are expert in these different fields, and we apply this broadly to aquatic species. First, a slide where I want to say something about aquaculture. I know that the talk was, the title says the genetic diversity issues in related to domestication. If we do domestication, then typically we're also doing aquaculture. So we're domesticating species to do well in the kept environment that humans provide for them. And if we look at aquaculture compared to other production traits, this is of course not news to most of you probably. It's relatively young. We see that in contrast to other animal production, often wild broodstock are used to provide production animals. Domestication is starting, but there's not much of it. And some of the major species, salmon, trout, tilapia, shrimp, sea bass, sea bream, they have genetic improvement programs. Several of them that operate worldwide. But I'd like to show this slide, which is a result from a project Fish Boost that ran 7 or 8 years ago, a European project, which did a survey and concluded that at least 362 fish species are farmed commercially. But if you look at the fish that are being farmed, most of them worldwide do not come from breeding programs. So in Europe, you see a high percentage. That's because we farm only a few species, mainly salmon and trout in the north, sea bass and sea bream, European sea bass, in the Mediterranean area. And for these, there are breeding programs, which means that we know the origin of the animals that we put in production, and also the origins of their parents, and we keep track of their pedigrees. In the big circle, which the size of the circle represents the amount of aquaculture production. So you see there's the majority of the aquaculture production is on the eastern part of the map. There, most of the produced fish are not from breeding programs or not from domesticated, not even from domesticated parents. Often wild broodstock is used for a lot of species. But this is changing. So we are seeing many more species being reproduced in captivity. So we see hatcheries of a lot of smaller production species. And then the question arises, what do we do about genetic diversity? So I'll address these 4 points. First of all, what is it and why is it important? And another question is, why do we need a strategy? Because, okay, in the wild, populations do not have a genetic diversity strategy. So why do we need one when we're doing domestication? And then the major part of the presentation will be about what can we do? So what kind of strategies can we implement? And I'll show some examples from programs that we started on sea bass and sea bream some 12, 13 years ago in Europe. And also another one that was started more recently, 2-3 years ago in Kenya, where we support a breeding program for a local tilapia strain. Okay, these are not fish. But I like this slide because it shows a lot of phenotypic variation. So it's one species. All these animals here, they belong to the same species. And I have this slide because both in our group and also in my past, before I joined Wageningen University, I worked on pig breeding, worked for a pig breeding company in the US. And I like this slide because it shows variation. And of course, this is phenotypic variation. This is variation that we can see. And then if we, but underlying these phenotypic differences are genetic differences, right? So if we, from one of these strains, if we produce offspring, they have more or less the same phenotypic pattern, which means that this is all genetically determined. So within the species, we have a lot of genetic diversity. Within breeds, this is a different story. So some breeds, they suffer from a small genetic basis and or problems with inbreeding. I'm not going to dwell on this very much, but so genetic diversity is variation at the DNA level. So how do we typically measure this? We can say, okay, how many SNPs, how many variant positions do we have per 1,000 nucleotides? We can also look at heterozygosity of the variable loci that are present in a species. So the second one is more useful for us when we do domestication, because this level of how many SNPs or how many variants there are per 1,000 nucleotides, this varies quite considerably between species and also between populations within a species. And it's not necessarily a measure of low genetic diversity if you have fewer SNPs per length of DNA. But the variable positions that are there, if we look at their heterozygosity, that is within species a more useful measure to look at the genetic diversity or to quantify genetic diversity. Then I also put here pedigree relationships, because in a lot of populations we don't take sequence or genotype information, but we use the pedigree. And I'll show in the second part of the presentation how using these pedigree relationships we can make sure that the genetic diversity that exists at the DNA level, how we can by tracing the pedigree and using the pedigree to select animals for reproduction, how we can maintain the genetic diversity, even though we don't know the actual level of it, but we know that whatever is there, we maintain by doing proper pedigree management, which is the most common way of managing genetic diversity. Okay, why is it important? Well, you will be familiar with this. If we have less genetic diversity, we are more likely to mate parents that are related to each other. And this leads to inbreeding. And so if we have a lot of genetic diversity, animals are from a lot of different families, then we avoid these inbreeding effects. So inbreeding can lead to reduced resistance to disease, reduced performance. So when we do domestication, often we aim to do production with these species. So we intend to use them for aquaculture. So we're also interested in their growth performance, for instance. And also usually the first sign of inbreeding is that we see increased deformations in the population. It's also important to keep genetic diversity to let the population adapt. So if we domesticate animals, we take them from the wild and we adapt them to our captive environment. So the environment that they have to deal with is changing a lot. So some animals are better at this than others, and that's in part also due to genetic differences between these animals. So if we change the environment, or if we have a changing environment imposed on us, it's good to have genetic variation for the population to adapt to that. So that's in the context of domestication. We also often want to do selection. So we want to improve certain features or reduce the occurrence of other phenotypes. So in order to do that, in order to do selection, we need to have variation. Otherwise, all fish are genetically the same. And then why do we need a strategy? Because in the wild, the populations take care of themselves. Of course, when they become under pressure, they have reduced population sizes. They may also get into trouble. But typically, if we leave the population and their environment alone, they do fine in terms of genetic diversity. We need a strategy when we do domestication because we have limited resources. We want to do as good a job with a limited amount of input. So we have a limited ability to source wild fish. There may be fish of a certain species that is a target for domestication in many locations, but in order to sample them from all these locations, it takes resources to go there, to sample them, to keep track of their origin, etc. Then domestication means we have to provide for the animals. So we have a space requirement and a cost for keeping all these parent fish in the subsequent generations. And then biology may be against us. So what do I mean with that? If we look at new species that are being held in captivity, often the new hatcheries pop up. They take fish from the wild and they start to provide, they do reproduction and provide fingerlings or fry for producers or for their own grow out. And in many fish species, fecundity of the female is very, very high. So we can get a lot of offspring from a very small number of parent fish. Now, if your aim is to provide fingerlings or to provide fish for your ponds to produce fish, it is tempting to do that from a limited number of parents, because parents, you need to feed them, you need to keep them, and so on. And if you can get like in catfish, you can get 80,000, 100,000 offspring from one female. Why keep tens or more than 100 female animals to maintain your genetic diversity in your population? It's tempting to only keep the animals that you need for your production purposes, for your the things that you do to create revenue from your population. But if we take all our offspring from a few females, we know that we will get a few large full sip families. All of these offspring will be non-inbred because their parents are not related. But in the next generation, we start to have problems, because if we have only a few families, then matings in the next generation will have a high chance of selecting two full sips, especially if we do not keep track of the pedigree. So what we can do is to produce many families, which gives us a small chance of selecting two full sips, even if we don't keep track of the pedigree. So in a lot of species, there's not a lot of resources to set up a control program. So it means that you can do some, you can take a strategy where you don't keep track of the families, but you set up a structure that ensures that you will have many smaller families, and you, by chance, are less likely to create inbreeding. This is an example of the start of one of the sea bass programs that we were involved in some 10-15 years ago. We had multiple groups of parents, and the peculiarity of sea bass is that they do natural mating in groups. So we have these parents, these 50 females and 25 males in one tank, and what we can do is collect all the eggs that are produced in one spawning event. So then, after the fact, we genotyped 300 offspring, 300 larvae with microsatellites. Remember, this is 15 years ago. And we found 95 diploid genotypes, and we reconstructed the parentage, because we had also genotypes on the parents, to see what happened in the natural mating. And this is again an example of where biology may be against us. So we have 75 parents in the tank, but we see that the offspring, they descend from only 7 females out of 50. If we look at the males, they have a higher probability of contributing, so 15 out of 25 contribute offspring. But in addition, you see that, okay, there is this male that has some offspring, but this male, he has 120 out of the 300 offspring. Then if we look at the combination, so if we look at the family sizes, we see that the biggest family here has 118 offspring of the same father and the same mother. So we have a few here. Typically, we have 5-6 large families that contribute 95% of the offspring, and then we have some smaller contributions. So even though we keep enough parents, we still may end up in the next generation with a few families, which then later on will lead to inbreeding problems. So we have a loss of genetic diversity because not all the parents contribute equally. So what can we do? We can, I mentioned already, we can keep track of the pedigree. So if we know that the parents that we use for reproduction have a sufficiently diverse background in terms of family, we can track that. So what we do is minimize the kinship, so minimize the relationship between parents that are part of a generation. We can use optimal contribution, so we can skew this L-shaped distribution of the individual contributions. We can change that if we know the pedigree. We can say, okay, from this big family, we take a few animals and we take all the animals of the smaller families. So that will help us to increase genetic diversity. But then we need to keep track of the pedigree if we want to use pedigree. So many of you are probably familiar with fish tagging. It's stressful on the fish. It is a lot of work, but quite necessary if you want to keep track of pedigree in fish. But it requires that the fish needs to be big enough to be tagged. So you need to produce families separately and keep them separate until tagging. And this is expensive if you need a facility like this. And sometimes it's biologically not possible if you have natural mating. This is another way of, this is a high-tech way of keeping families separate. It can also be done in ponds, of course, but you have to set up something like 50 or 100 small happas and control, have very good bookkeeping of who is where and so on. So with pedigree, we can do it. If we don't have pedigree, we can do genotyping, as I showed earlier, but then we are relying on what we will find once the fish are big enough to be genotyped and tagged. So we can also use strategies like migration and rotation. So we can, what it does is subdivide the population in more groups. So if we have one mating group, like I showed earlier, with 50 females and 25 males, we can also have 5 groups with 10 females and 5 males each. And that means that, okay, offspring in one group, they are not full SIPs. We know for sure they're not related to, closely related to animals in another offspring group. This is a slide explaining how we can do reconstruction of relationships with DNA markers. I think I don't need to explain this very in much detail to this group. So we genotype the fish and we look in the offspring if they have alleles that come from certain parents. So we can decide that these two fish are full SIPs in this case. What I was explaining is that we can, if we make subdivision of the population and we do a migration between groups, we can do something that's called a rotation. So if we look on the left here, if we have 4 groups, 1, 2, 3, 4, and in generation 1, we take, we make offspring, and then the offspring, we take, for instance, the males from group 1, and we group them together with the female offspring of group 2, and the males from group 2, we move them, the males from group 2, we move them to 3, and so on. And again, we do this, we get 4 offspring groups, and again, we move from the offspring group, we split them, females stay in the same group, and the males, they go to the next group. So if we do that, now we have to make a choice. Okay, how many groups do we divide our population in? Do we use only 3? Do we use 6, 4, like above? Or maybe we split it up into 20 different groups. So there is an optimum here. But this graph, it's a bit of a busy graph, but I'd like you to look at the blue line, which indicates if we have in total 48 males and 48 female parents, and they mate in single pairs. So this is not for a group spawning event. But if you do individual mating and then you group the offspring, so after you have the offspring, you don't no longer know their individual pedigree. If you look at the blue line, and it starts here at one group, putting everybody together, we have an average inbreeding rate, so the increase on inbreeding of 1.1. And if we then split up into 2 groups, 3 groups, 4 groups, 5 groups, you see that this inbreeding per generation, it goes down. Later, if we split it up, if we would split it up in even more groups, we have very small groups. So there are very few animals to mate with within the group. So then the inbreeding rate goes up again. So you see there's an optimum around between 5 and 10 groups. And if we do this simulation for different starting points, so for different size total population, we always see that the optimum comes out somewhere between 5 and 10 groups in the rotation. Okay, I've in the final part I have 2 examples of how we got started and what we did. So how to get started? First of all, you want to collect fish from multiple sources, so multiple farms or multiple lakes or multiple streams where this same species is present. So this is a plot after genotyping sea bass, which were coming from 14 different farms in the Mediterranean, each identified by a different color. You see that the genetic distance between these individuals, which is indicated in this 2D plot, you see that there is a grouping. So fish from the same farm, they tend to be more related to each other than fish between different farms. So take fish from many different origins. We see the same analysis was done for sea bream, and we see the same pattern. So in the initiation phase, we have to select parents to start the population. So select the founders. So take them as widely as possible. What we did in one example is in Kenya was we took fish from an existing hatchery population with some known, some unknown origin. And so the first thing we did is we want to know these individuals. So after sourcing, starting our source population, we take a subset of them, in this case 300 females and 60 males, and we already used, the aim was to set up a breeding program. So we already selected them based on size, and of course we need males and females. So those were the selection criteria just based on the phenotype. Then we measure the genetic diversity by genotyping and select again. So we genotyped them, we did the genetic diversity analysis, which resulted in a plot like this. So this is a heat plot. Basically, this is a relationship matrix, and dark colors indicate closer relationships. So you see on the diagonal the relationship with of the animal with itself. It has a dark red color, but everything else is quite light colored. So we were lucky here to find low genetic relationships between the animals. You see some grouping. So you see here a block of fish that were from apparently were descending from a certain origin or were related to each other for some reason. But even in this block, the relationships were not extremely high. So then out of the 360 animals that we genotyped, we optimize a subset of parents, and we do that by taking these values that are in the relationship matrix. I'll show if I have time, but maybe I'll go over that quickly. We use the relationship of an individual. So each row in this matrix is one individual, and it has a relationship value with all the other individuals. So we quantify if it's with a full SIP, your relationship is 0.5. You share 50% of your genes. But if it's a more distantly related individual, we have lower relationship values. So we found that on average, we see some very low relationships. So 2%. And like I said, if you have two full SIPs, a brother and a sister, or a brother and a brother, or two sisters, they share 50% of their genes. But on average, two fish from this population, they share 2% of their genes. So that is a measure of their genetic distance. If you take fish at random and you mate them, then the inbreeding in their offspring is half the relationship between the animals. So if on average, two random fish have a relationship of 2%, the inbreeding in their offspring will be 1%. And this is still within the limit that is suggested by FAO to have a maximum of 1% of increase in inbreeding per generation. So based on this average relationship of every individual with everybody else, we took animals that are here at the bottom of the distribution. So they're very different from everybody else in the population. These animals we avoid, they are on average much more closely related. Still okay, 0.07 is not that bad from experience, but still we have a choice. So we don't take these animals that are close related to the others, but we take from the other end of the distribution. So this is the explanation of how these values in the matrix are calculated. I can go over that, or we can do it in another session, but if I'm looking at the time, I should get closer to the end. So the additive genetic relationship, the 0.5 between full SIPs, it gives an inbreeding coefficient of 0.25 if you mate them together in their offspring. So the kinship coefficient is half the relationship. It's the predicted inbreeding in the offspring. So this is a pedigree, and the yellow matrix that I showed you, it's a graphical representation of a graph like this. So on the diagonal, you have the relationship of animals with itself, and on the off diagonal, you have the relationship between different animals. And to calculate kinship, we divide all the values by 2. And then in each row, you take the average. So the average of this individual in this pedigree, so individual 1, has an average relationship to everybody else in the pedigree of 0.6, including itself. And so it means a mean kinship of 0.3. If you do that for all the animals, then you can rank them on this value and you can choose the best ones. Then also in mating, you can avoid matings between closely related individuals. And that way you also for the next generation, you create less related individuals. So you can postpone the accumulation of inbreeding. One disadvantage of this approach is that it does not account for the high kinship in the beginning. So you have to make sure you start with a varied population. This is to manage the genetic diversity going forward. And if we select based on the average, we assume that all of these selected fish are going to be contributing equally in the future. And this is, depending on the mating system, is not always the case. So one of the last slides. So what we can also do is to optimize, and there's more sophisticated algorithms for this. We optimize. So here we say we have the mean kinship of an individual. If this value is low, we can do more matings with this individual compared to an animal that has a high mean kinship. And there are algorithms, optimal contribution algorithms, that will optimize the selection of animals given a certain threshold. So if you say, I want my mean kinship or my average relatedness to increase by a maximum of 1%, then use other criteria like growth rate or size or other important features to select fish, but I want you to keep this limit of maximum increase of 1%. And so this is an output of the breeding program that we run in Kenya. These are the different families. There are 50 families that we can choose from, and you see different numbers of animals being chosen from different families. There are a few families here that have contribute one animal to the next generation. Usually this is because they are genetically diverse, but they're not extremely good in the production traits that we are selecting for. This is probably a family that has very good growth rate, very good survival, and the other traits that we select for. So we select a lot from them, but in addition, we also use these animals that maybe are not the best, but we need them to maintain genetic diversity in the population going forward. So, take-home message. It's important to have a strategy from the beginning. So if you do everything right, but you've started out with very few individuals or very closely related individuals, then you'll still run into problems later on. If you have a good starting material, but reproduction causes very unequal contributions, you get into trouble later on. So start with diverse origins and know what you start with. So genotype the founders to know how at the genome level, how related they are to each other. Adapt your plan, not your plant. Adapt your plan to the reproductive biology and reproductive behavior. And you want to have a strategy from the beginning because I sometimes hear, oh, but if we need more genetic variation, we can bring in fish from the wild later. But that means these are fish that are not domesticated. So domestication is a selection process. Every generation, the population adapts better to the captive environment. Or if you're selecting for growth rate or other production traits, they are improved based on the wild population. So if you have to bring in genetic diversity later, it means that your efforts in domestication and selection are diluted by the wild fish that you're bringing in at a later stage. So, thank you. I'll be happy to take questions now or maybe after the next presentation. I'm not sure what the schedule is, but I'll I'll be happy to answer. Thanks.
Thank you, John, for this presentation. So this is very important for the basis to start the domestication. Yeah, I forget to mention that I will also give a short talk about the what we did during 5 years in the Netherlands, and then afterwards we will have a question session. Of course, I have a lot of questions for the presentation, but later then after I present my
Sure.
short talk. Yeah. Thank you, John. Yeah. And for everyone, if you have a question, please just put your question into the chat box, and maybe we can answer one by one. Okay, I will also start my presentation today. It's coming. So yeah, it's quite familiar with you, John, with this cover. So then, yeah, I will start today my talk related with what I did during my 5 years in the Netherlands. So this is my first chance to present after I start in January to work. I mean, to back to my institute in the Research Center for Applied Zoology. Even I moved from the ministry at 2022, and then during my study in the Netherlands, but then now I'm here at the Research Center of Applied Zoology. So my presentation today is entitled Breeding, Forage, and Growth in Tilapia. So I start with the so I start with the stressors in aquatic fauna. We know that in aquatic fauna, especially in the captive condition, in the controlled environments, we have stressor including the natural or environmental factors. So, for example, we have quite diverse of the aquaculture system that can also introduce natural or environmental factors. The next one is social factors, is including the density, so the correlation between the individual, the predatory and predatory correlation between the animal. It's also introduced the stressor, and also the artificial. Of course, if we culture fish in the captive condition, then we will intervene the fish as well. And then what the stressor affect the growth. So the first, if fish they get stress from the environmental, even the natural stress, social or the human intervention factor, they will reduce the feed intake, and then they will also reduce the vision, and then at the end of the process, they will affect the growth at the end of the process. And then in this study, we aim to measure the resident growth. What is the resident growth? We define the resident growth as the natural logarithmic of variance of deviation. So in this slide, I will give you a short preview about what is the resident growth. So if we culture fish during the grow-out period, we can take the records of body weight, multiple time points. So in this case, the y-axis is the time points, and the x-axis is the time points, and y-axis is the weight. So then we can measure weight several or multiple time points. So for example, at time point 1 until time point 4, indicate with the fish picture in this slide. And then from the record of the multiple weight, we can also estimate expected growth. In this study, we use the nonlinear regression to estimate the expected growth. And from the expected growth, we can calculate deviation between time points. So each time points, we have deviation between the expected growth and then observed weight. So then after we have deviation between time point, we can calculate the variation of deviation. The smaller deviation means that fish they can cope with the stressor, so they have more consistent growth. It means they can cope with the stressor. And if they have a bigger deviation, they have less consistent in growth, and it means they disrupt by the stressors. And then we can increase or we can select fish with the consistent growth using the selective breeding. So in this study, we use the fish population of gift tilapia in Malaysia. So normally, traditional farmer, they will culture fish in the non-aerated ponds. So traditional ponds, they don't use any aerator. So they will this non-aerated pond will introduce the stress factors of the dissolved oxygen fluctuation, and then we measure the daily dissolved oxygen fluctuation is 2 under 7 ppt, so it's quite fluctuate daily. And then we measure weight several time points, 5 time points, and then we calculate the LNFAR from this population. In the second study, we used the Sukamari tilapia. So we introduced the monoculture and co-culture shrimp. As I mentioned before, that the co-culture with shrimp also introduce stress in this case, because the relation between the species in the system, they will could be disrupt the growth, or maybe disrupt the performance of tilapia. So in this case, we use monoculture, aquaculture, and shrimp. And then we also measure that there is a salinity fluctuation, 625 ppt, and also in this case we use the traditional ponds, and we measure the dissolved oxygen fluctuation is 206 ppt. So quite a lot of distraction here in the second step. And the result showed that the LNFAR, as I mentioned, the growth consistency, they more heritable. The heritability, first I need to mention that what is heritability? The heritability is the percentage of genetic merit that can be inherited to the next generation. So in this case, we measure the heritability of LNFAR and also growth. Growth is the most important trait in aquaculture breeding. So then in these 2 studies, we had the result. We have the result that the LNFAR is more heritable in the environment with more stressor. And then for growth, they have the comparable heritability between environment with more stressor and then less stressors. And then the next question is, resilient growth is important. So then in the next study, we calculate the economic value of resilient growth. We calculate the economic value of resilient growth based on how fish growth, and then related with the feed waste, growth efficiency, and feed safe. In 10.2, we can see that if fish grow below the expected growth, we can have feed waste, because if we feed fish ad libitum, but then we feed based on the expected growth, then we have the feed waste. At 10.3, if fish they grow above the expected growth, then we have a growth deficiency. It means that we can feed. I mean, they have the potential to grow until the specific, until the observed growth, but we give them feed at the expected growth level. So we have a growth deficiency, but in the other hand, we have a feed safe, because we don't give feed to the fish. And then from this calculation, we calculate that the economic value of LNFAR is $0.3 per unit of LNFAR per kilogram. In the next part of this study, I did a simulation of tilapia in the tilapia breeding program. So I simulate 3 scenarios. The first scenario is social, the second scenario is economic scenario, and then the last one is the environmental scenario. In the y-axis is the selection response for growth, and in the x-axis is the selection response of resilient growth, or the growth consistency. For example, if we take the green button at the bottom of this image, we push fish that we select fish that they have the lowest LNFAR. It means that we select fish that more environmental friendly, because they will have less feed waste, and also because they have the less feed waste. And then if we select fish in this specific area, and then we will have a reduction in the selection response of growth. But for example, if we select fish based on the economic scenario, in this case, the economic scenario, it means that we push fish to growth as we select fish with the best growth performance, and they will reduce the LNFR, and then we will go into the economic scenario. And for the social welfare, we still uncertain about whether fish with low LNFR or high LNFR have the as the indicator for social welfare. So we put the LNFR as the constant. But then if you put the LNFR as the constant, and then they will reduce the selection response in growth. As the take-home message, so the resilient growth is moderately heritable, and then resilient growth is a sensitive measure to test G by E for growth. Resilient growth is more predictable, which leads to cost saving, and then the positive economic response is expected from improving resilient growth. So this study, I defended my thesis at the 4th of December last year. So with the title of Breeding Forest in Tilapia. I would like to thanks to Wageningen University Research, PDP, Coupon Shifting, Ministry of Marine and Fisheries, World Fish, and also National Research and Innovation Agency. This is my first part of the talk, John, and everyone. And then I would like to update what I did during 8 months in Research Center of Applied Zoology. So we are now in the focus group of fish genetic and reproduction. So mostly we did fish domestication, and we have several researchers in contribute to this focus group. So most of the research based on the population genetic for domestication, fish reproduction, and population management with genetic approach. I will come back to this part of research later. So this is mostly for ongoing research that we did in a focus group of aquatic fauna. First, we start the domestication and genetic improvement of Hemibagrus nemurus. Is a catfish family. So then the next two part is related with the endemic fish species in Bangka Belitung. So it's a Beta burdigala in Bangka Island, and then we also have a chance. We get the funding to do the whole genome sequencing for Bangka Island endemic fish. And the last one, we had the collaboration with the foundation, and then we initiate to have a research station for domestication and genetic improvement for prospective local fish, including catfish, torsoro, and serak. So this is the disperse of our research ongoing in our focus group. First, I would like to talk about the Hemibagrus namurus. So Hemibagrus namurus is the part of the catfish family. So this is part of collaboration between our research center, and then the agency of fisheries in the Central Java. Due to the human activities, excessive exploitation, and habitat discontinuation, we have a population fragmentation, decreasing population, also decreasing genetic diversity. So then the aspiration from the fisheries agency of Central Java, they would like to have the domestication program and also the genetic improvement for this species. The next one, we also had the aspiration from our collaborator in Bangka Belitung Island. So it's in Sumatra part of Indonesia. So Beta burdigala is critically endangered species and also endemic species in Bangka Belitung. Last month visit to the habitat of the Beta burdigala, we had a quite big destruction of their native habitat, because the expansion of the tin mining, and also for the palm crude oil expansion. So these two species, I have the idea to have the fish domestication model, And then for non-food species, I have Burdigala as the model of fish species. And then for the food purpose, we have Hemibagrus nemurus for the fish model. And then what is the invention of this model? Because then the normal business of the fish domestication is the only to characterize the population, and then we do adaptation and the production, and that's it. We thought introducing what you present today to introduce the genetic approach and to maintain the diversity and etc. So that's the idea to do this program. And this program is at the very initial stage, because then we still need to find additional funding from the agency, and then also we still need to start the program. I think next year we will start the program for Hemibagrus nemurus, and then for the Beta Burdigala, we had ongoing project because we have funding with the Ministry of Education and also with the several foundation. And then the next one is we had a call for funding for whole genome endemic fish from the Bangka Belitung Island. So this is the chance to use the most recently sequencing technique, because I think this is the first PECView review machine, the first PECView review machine in Indonesia, and then we had the chance to do the whole genome sequencing for the 4 species in Bangka Belitung, including Beta Burdigala, Chlorofaring, Parus pronemus, and then Encheloclarias. And the last one, this is already initiated by our colleagues in the Center of Applied Zoology. Otong and Jojo do initiate this research station for prospective local fish. So we have 3 working package related with local fish. So the first one is research and development of local fish, including catfish and also taurus. I think we will start with catfish and also the carp. The next one is research development of Sera quadricarinatus. So it's not native Indonesia, but we have aspiration to start research on development of this species. And then the last one is research and development of Torsoro. This is also in the start, the start of the initiation program, and then we already built the broodstock maintenance facility in Anyar. So it's West Java, is 2 hours from Jakarta, and then we also had the multi-species hatchery for this for this research station. Yeah. So that's it, the ongoing research that we are doing now. So I would like to thanks to our collaborator that I think they also joined this session today from the Bangka Belitung University, Research Agency of Central Java, Matahati Foundation, YSDS that sponsored by Paninbank, and also Ministry of Education, Gajah Mada University, Erlangga University, and also IPB University. So thank you, and that's it, my presentation for today. So then now I will come back to another role as the moderator. So if anyone has the question, please raise your hand, or maybe we can start with the chat. If anyone has a question, so basically we had a hybrid session, John. Here we have people like 30 people joining offline, and also we have 65 participants joining online, and we will also stream at YouTube now. If anyone from this room, if you have a question, please raise your hand. Otherwise, I will start with the question from the chat box. Okay. Okay, maybe we can start with the question from the chat box. First question from the Adinda Jacinda. Question for John. How can the success of domestication and selection be evaluated for establishing a base population, particularly from wild captive stock, before entering an existing breeding program? And how long does the process take? or what parameters indicates that it has been successful? Please, John.
Yeah, thanks. It's a good question. So there are several questions there.
Yeah.
So let's say if we want to do domestication and selection, because I have a comment on that after your second presentation, Farid. If we want to do domestication and selection, how can we conclude that we are successful? That is what the question is asking. Well, the first thing that we have to be successful in is reproducing the required number of parents. So reproduction success is the very first measure of success. Now this varies a lot. So some species, you just put them together in an aquarium and you get offspring. We also have a research project already for more than 10 years here in Wageningen to reproduce European eel in captivity. And this is slowly progressing and taking a lot of effort to even get live fish offspring. So that's the first measure of success. How many parents are contributing to next generations? So there's some rules of thumb that you want to have a effective population size is a population genetics measure that is at least 50 and preferably 100 effective parents. So that's weighting their contributions, weighting their relative contributions compared to a random mating population. So if you can keep track of the pedigree, you can measure, am I using and am I getting offspring from a sufficient number of different parents and lowly related parents each generation. And so you can trace basically the increase in kinship or the increase in future inbreeding. That is a key parameter to keep track of. That's when it's established. And let's see what parameters indicate that it has been successful. So this inbreeding rate is a parameter to say that the maintenance of genetic diversity has been successful. Whether there's a lot or little genetic variation from the start. One measure we can use is if we, in addition to pedigree, also measure some phenotypes. So especially if fish are domesticated for breeding for production, we measure harvest weight or growth rate. And with the pedigree and these phenotypes, we can estimate the genetic variance or the heritability. It's a more common parameter. And we know that heritabilities for harvest weight and for growth rate, they are for genetically healthy populations, they're typically in the range of 0.3 to 0.4. Now, if you have low genetic variation, then these values go down. So estimating after 2 generations, you can already make these estimates. Estimating the heritability of some of these common well-known traits, you can compare to a reference value of around 0.3 to 0.5 that you, 0.3 up to 0.5. So 0.3, 0.4 is what you expect to see for a genetically healthy population for these heritability measures. So inbreeding rate and heritability of some simple phenotypes, those are the key parameters, I would say. Did that answer the question? Great. One comment I want to make after your talk, Farid, in my talk, I focused on programs that domesticate populations that will stay domesticated and will be used for aquaculture. if you do conservation, you probably want to limit the amount of domestication, or you want to not adapt the species to the captive environment. If your aim is to at some point reintroduce these species into the wild, these fish into the wild, you want them to still be adapted to the wild environment. So then there's a different so then you have a tension between, okay, they need to be domesticated enough so you can reproduce them effectively and you can keep their genetic diversity, but you don't want to change them too much from their adaptive state to their natural environment. That's for if you want to breed them for restocking purposes, that's an additional thing you have to monitor. So you want to probably keep track of is their morphology changing, or you want to make sure that the morphology is not changing. You want you mentioned predators at some point, presence of predators. Yeah, behavior in response to predators that can disappear if you do domestication.
Yeah.
So you need additional effort for that if your aim is to reintroduce to the wild.
Yeah. Of course, we have two models for the domestication that I presented before, John. So I think that the discussion on when we want to restock the fish is related with the fish that we had in Bangka Belitung, so it's Betta budigala that critically endangered. Maybe I will take that suggestion that yeah, of course, we don't want to lose their their, for example, their specific characteristic of the morphology that they can adapt to the different. But for the Hemibagus nemurus, of course, we people are tend to use as the aquaculture production, but then yeah, that's also the difficulties if they want also to use as aquaculture sources, I mean the food sources, but then they also want to increase the number of population in the river. So maybe so if you have any suggestion, John, maybe we do this like balance breeding, but I don't know what is the balance breeding if we want to use as food sources and also to do the stocking burst, because in my opinion, the balance breeding is to balance between two traits. For example, if we do the dual goal of the selective breeding. So yeah.
No, balance breeding is good, but it also means you're making a compromise between two different efforts, two different goals. Yeah, there is a lot of discussion also in Europe, of course. On we have a lot of salmon production in Norway. There's been some studies on how do the escaped fish, because salmon is truly selected for aquaculture production. How do the escaped fish affect the genetic diversity of the wild populations? So it's not an integrated goal from the beginning. I think in your case, you already are aware and you already have these dual purposes of wild restocking and aquaculture with your breeding program. So you're one step ahead of that issue. But how to do it optimally, that's indeed still an outstanding question.
Yeah. Okay, so yeah, before so is there anyone that want to have a question here in the rooms? No one? Okay, then we continue with the second question. Oh, okay. Are you online as well?
Yeah.
Okay, yes, please. So there is a question from Sendra Pahan here offline.
Thank you, Mr. Farid. Okay, I have a question to Bastian. Thank you for your presentation. I'm sorry if I didn't get your presentation. to maintaining genetic diversity, you say avoid low management mean kinship matings. And what do you mean avoid low mean kinship matings? If we have one population in one plot.
So yeah, I didn't explain that. It was on one of the slides. It says so I said avoid mating between close relatives. That I think is more clear. But also avoid matings between individuals that are where one individual is closely related to everybody else in the population, and another individual has very low relationship. The reason is that if you have an individual that is quite unique in the population, you want its offspring also to be as unique as possible. If you mate that unique individual, so this could be also an individual that you take from outside, if you mate that individual to let's say a parent that has a lot of offspring in the current population, then those offspring, they will get half of that close relationship to the current population. So they immediately move up in this. You remember that distribution of how individuals are related to the total of the population. They immediately move up to the middle, and you can only you reduce the amount of fish that are at the lower end of the population more quickly if you do it that way, compared to mating lowly related individuals to an average, so to some highly related and some lowly related individuals. So you spread their genes more evenly in the population is another way to look at that. So in the long term, you have more benefit from an introduced fish if you mate it to different animals in the population compared to mating it immediately to the most close related or the most inbred animals. So it's a long-term effect that you, long-term benefit that you get if you avoid these high kinship with low kinship matings.
Thank you. Is that answer the question?
Yeah, thank you, Dr. Bastian. So when should we rotate or migrate a population or group to maintain genetic diversity? Mic-nya belum hilang.
Okay, it's still mute.
Thank you, Dr. Bastian. So when should we rotate or migrate the population or group to maintaining genetic diversity without we have a genetic testing?
Right. So when is it useful to do rotation? It's the lowest input approach, I would say. So if you don't have resources or facilities to keep track of the pedigree, so if you have a generation of offspring that you know is produced by a certain number of parents, but you don't know the individual, the parents of each individual, if that is your situation, you cannot afford to genotyping, or there's no technical ways to do tracing of pedigrees, then the rotation, so the grouping and rotation, is the lowest tech approach that helps you to avoid long-term inbreeding in the population.
Thank you, John. Thank you, Suri. So I think your the last answer related with the lowest the lowest standard to maintain the genetic diversity is also answer the. Actually, I have a question for the beta species, because the fish is quite small, and I think it's impossible to tag the fish because they're quite small. And then I think yeah, the idea to do the rotation mating, I think is applicable for this kind of species. So yeah, so that's and yeah, and then another question, John, to you related with your presentation. So if we have several sources of several genetic sources population as the candidate for the domestication, should we check the genetic diversity per population? So that's the question.
Yeah, good question. And it's important, especially if your aim is restocking. So if your aim is to create a population that will stay domesticated in captive environment as one population for the future, then the total genetic diversity is what's important to you. But if your aim is to do restocking or to serve communities in different river systems that may want their local population in their production environment, then yes, you want to know how different they are and whether you should even mix them. Now, deciding when it is okay to mix them or not is more often a financial decision than a genetic decision. But it is important to know, especially if you want to send fish back to those different locations.
Okay, thank you, John. We still have one question from the chat box. Perfect to me. What are the approaches to trap breeding that can be done to be resistant to disease and breeding for all male monosex or sterile triploid? Thank you. Can we get the percentage method? Okay, then we will get back to you. So I think yeah. So I think your question is also answer your question also, because then this is what the lab breeding did that can be done to be resistant to disease. So we can do the breeding program with the breeding goal to have resistant to disease, but of course not only for resistant to disease, but also maybe dual purpose with the growth, because growth is still the most important trait for the aquaculture. And then breeding for all male tilapia and or sterile triploid. So yeah, I don't have any update yet about breeding for all male monosex or sterile triploid. Do you have any suggestion or answer for this question as well, Jen? If you have some update on that.
Yeah, breeding for all male would be really interesting. So what has been tried in the past is to cross blue tilapia and black tilapia, so subspecies of tilapia. The idea was that the different species would have a different sex-determining locus or sex-determining gene. So one would be XY, and the other one would be ZW, like in birds, chickens, for instance. But this has not led to clear success that you would have genetically or genetically all male development. So the standard in tilapia production is still to use chemical treatment to create all male. But indeed, there has been effort to look at this sex determining locus, and the other option is to create what's called the super males. So that only give single sex male offspring. That I'm not a reproductive specialist, but I understand that could work, but it would require these super males to be created in an efficient way. And sterile triploid, I haven't seen triploid. I've seen I've seen efforts for developing sterile tilapia, but not by triploidy. This is by a switch on a sex development gene. But yeah, also have to forgive me for the details. But there is research effort to produce sterile tilapia offspring.
Okay, thank you, John. I think this is the last question. So from the chat box, Andy want to ask a question. Are you still there, Andy?
Halo,
Please.
ya. Suaraku kedengeran nggak ya?
Yes, we hear you very well. I think Andy is the master of animal breeding genomics in animals. He's graduated from one. Please, Andy. Halo, Andy.
Halo, suaraku masuk?
Masuk.
Okay, John, long time no see. So I just want to give a follow-up from your first answer to Ashinda that your answer about the parameter that can be indicator for the successful of the breeding. I remember that it's quite important to set up the breeding goal first. That's why there is a question appear from you to also to Mr. Farid about what he's doing right now. Is it conservatory program or is it for the farm or from the domesticated one? Because I think it's quite important, especially in Indonesia, if we start first with the breeding goals first. So in which part that we are going to go, so we can measure our progress in our breeding through the breeding goals. That's my comment for your answer, and also for Mr. Farid about your programs about the breeding of beta in Bangka, and also the Torsoro, and also the Surax. I follow all of your work, and then I think we need to be clear first about the breeding goals, in which part that we are going to progress in genetically.
Yes,
That's
right.
my.
It's a fair comment. That is the first step. In my answer to Adinda Jacinda, I focused on the evaluation of the let's say the aim of genetic diversity. So breeding programs have two aims. One is selection, genetic improvement, and the other is maintaining genetic diversity. And so my answer focused on maintaining genetic diversity. How can we make sure of that, and how can we measure that we are doing a good job? And you rightfully address the other of the two goals of breeding programs. This is to make progress, or to maintain the certain phenotypic levels, and for that you need to start by defining that first. So where do you want to go, or where do you want to stay? So that's the breeding goal. Yes. Yeah. Very
Okay,
good.
and I have a question for you, John. So in Indonesia, we as a fish farmer, also our offsprings also by some by government or by and also NGO by also our offspring to restock in the wilds, in the river, everywhere. So if we can maintain the inbreeding below 1% or below 1%, is it okay for us to release our fish to the wild or to the river or everywhere, or we need other considerations? So from genetic perspective, that we can sell our fish to the NGO or to the governments for them to release to the wild. That's my question. Thank you.
Yeah, so it relates to the first question, right? So if the fish are suitable for release, I think that is what is the main question. So yeah, I think if the government or NGO is buying fish for release, they should ask for or assess themselves if these fish are suitable for release, or if they will be good for the population, or if you have fish from a single family and you release them to a system, a lot of them, yeah, you may actually you may actually not be doing the population a favor, and that's very underemphasized. So typically restocking is focusing on numbers of fish and not so much on or hardly at all on the genetic diversity or the genetic match of the fish that you're stocking to the natural population that is already there. I have to go to another meeting that's just started. Farid, is it okay to round off the discussion here?
I think so, John. That is the last question, and then we can end the discussion. And then, yeah, of course, we still have a continuation of the discussion related with my ongoing research, our ongoing research in zoology. So maybe we can find another time to have a further discussion related with the.
Yeah, I'm perfectly happy to discuss with you or others in the audience that if you have further questions, contact me, and just we can go into much more detail if you want.
Yes, of course. Yes. So yeah, I would like to thank very much to you, John, for your taking your time to talk and then to answer the question for the discussion. So then, yeah, so with this last question, so I ended the discussion, and then, yeah, I will send back the session to the master of ceremony. Yes.
Baik, terima kasih Mas Farid atas bantuannya hari ini untuk menjadi moderator. Dengan begitu pula juga berakhir acara kita untuk hari ini. Bu Kapus, akan ada closing remark? Tidak? Oke. Baik, terima kasih. Sebelum kita selesaikan, before we close this event, John, we want to take a picture together. So, would you mind everybody from the participants to open the video so we can have a good picture together?
Okay.
We have 3 slides, and I will start with the first one. Oh, I'm sorry, I haven't started my screen yet. Yeah. Okay, 1, 2, 3. Okay, and the second slide. 1, 2, and 3. Okay, and the third slide, the last slide. Okay, finished. Thank you very much, everybody. Thank you, Dr. John Bastian, and thank you, Dr. Farid, for today's. I think this event is already over, so we hope to see you soon in another chance. Thank you very much. Terima kasih teman-teman untuk join di acara ini. Kita bisa ketemu lagi minggu depan di acara Summer School dan acara-acara lainnya yang ada di Zoologi Terapan. Silahkan bisa follow di sosial media. Terima kasih. Saya mohon maaf kalau ada kekurangan. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, John. We can run off the demo.
Thanks, everyone.
Yeah. See you. Bye.
Till next time. Bye-bye.
See you next time. Bye.